I'm going to be talking to you about gene therapy and specifically our gene therapy. So I'm sure you're aware that gene therapy has become a very, very important modality in healthcare right now. It's a way to provide lasting cures to people who have genetically defined diseases and even some other diseases. Yeah.
Before we jump into it, can you just introduce yourself?
Oh, sorry.
How you got here and what gene therapy. You've got a very intriguing deal.
Yeah. So yeah, my name's Don Haut. I'm the CEO of Carmine Therapeutics. My background, I was a molecular biologist ages and ages ago who learned during his PhD that the most important thing in my PhD was I didn't want to be a molecular biologist. Yes. What about this microphone as well? Much better. Sure. Yeah. I mean, it's like you're in the ocean back there with the blowers. Yeah, I did an MBA after that. Went to McKinsey and Company. Have been in biotech and other healthcare companies ever since. Most recently, before this, at AskBio, which was another gene therapy player that we sold to Bayer for about $4 billion. Anyway, so back to Carmine Therapeutics. So Carmine, we are a gene therapy company as well, but we have identified or were started on the basis of the notion that gene therapy is important, but there are a lot of challenges with the two most available platforms for delivery right now, that being viruses or lipid nanoparticles. They all cause significant inflammation. They've got dosing challenges in terms of you can only dose the viruses once because you build up an immune response to them. They also have size restrictions on what you can put in them. At Carmine, can I have the next slide? At Carmine, we've decided to take a little different approach, and we use, next slide. Okay, thanks. We use extracellular vesicles to deliver our genes to the target cells. So it's been learned over really the past 30 or 40 years that all cells in the body shed and take up these extracellular vesicles from each other. And it's a way for cells to communicate with each other and coordinate their activity. And can I have the next slide, please? What we do is we start out with red blood cells. So donated red blood cells. And specifically, we're using O-negative red blood cells. And just to remind you, O-negative cells donors are the universal donor. They can be used in anybody and they can be redosed in anybody. We started out with these and we use them, we make extracellular vesicles from them. We can make massive amounts of extracellular vesicles from donated human blood. We can then load them with DNA or RNA or other nucleic acids and then deliver them to animals. Have the next slide, please. And what you get with these extracellular vesicles, we have very, very high amplitude and very long-term transgene expression. I can show you some data about that. They're redosable. They can carry enormous payloads as well as multiple kinds of payloads. And they are targetable and can be used in multiple different cell types. Can I have the next slide, please? So here's a little data for you. And what you can see here in this data is on the left-hand side is the transgene expression, and the top line there going up to 18%. This, by the way, is factor 9, human factor 9, which is important in hemophilia B. That is our production of human factor 9 there. We can get up to 18% of wild type, which is well into the therapeutic range for this particular protein. The other line you see down there is 6%. 6% is the highest that one of our biggest competitors has been able to do, and that's Generation Bio. And anybody who follows gene therapy has probably heard of that company. On the right-hand side, and I don't know if it's on your screen too, but it's partially blocked on mine. On the right-hand slide, you can see a little bit of, thank you. You can see durability, and we can get transgene expression out at least a year and a half. This is an ongoing experiment, and we're still monitoring. Can I have the next slide, please? I mentioned that we can get genes into lots of different cell types. We can get it into the lung. You can see on the top left-hand panel, we can get them into the back of the eye and the retina. We can get them into the liver in the bottom left-hand panel. And brand new data, we can actually get them into the brain as well, which is really exciting. It's something that's so new for us, we honestly haven't even figured out what we're going to do with that one yet. Can I have the next slide, please? So this compares our platform to the other platforms, to viral and lipid nanoparticles. Viral really is the standard right now. You get excellent transgene expression. Lipid nanoparticles, you get a little bit, you get about, you don't get as well, and we're located there about halfway in between the two of them. We are far safer than either viral or lipid nanoparticle because, again, we're taking advantage of a natural process that occurs in your body all the time. In terms of redosing, we can redose, whereas lipid nanoparticles can't, or sorry, whereas viral can. In terms of payload, we have the best payload capacity of any, hands down. And in terms of manufacturing, pretty sure that's the bottom, we are easy and probably 2 to 3 orders of magnitude less costly to manufacture than the viral systems. Can I have the next slide, please? So what are we going to do with this? Right now, we're working on several different programs. Initially in the back of the eye, we're working on Stargardt's disease, which you may not have heard of, which I'll go into in just a moment. We're also working on macular degeneration, which chances are you have heard of. Just because if you live long enough, your chances of getting macular degeneration are pretty good. We're working on cystic fibrosis. The Cystic Fibrosis Foundation is actually one of the cornerstone investors in the company at this point. We will begin working as well on primary ciliary dyskinesia. And we have 2 programs ongoing with Takeda as well that are confidential. And the next slide, please. So just a few comments about our work in the back of the eye in ophthalmology. We're working again on Stargardt's and age-related macular degeneration. Next slide, please. So these 2 diseases are actually pretty similar. They have a different gene and different mechanism involved, but they both lead to blindness that kind of works like this. Starts in the middle and works its way out. The difference being that Stargardt's disease is a genetically defined disease that only happens to people who have a bad copy of a gene called ABCA4. And Stargardt's disease generally makes people blind in their teens, whereas macular degeneration has a different cause and generally leads to blindness in your later years. Can I have the next slide, please? So I'll start out with why we think we can do this. So one of the things that I mentioned before is that we are able to deliver these genes to the back of the eye. And what you're looking at here is an image on the left-hand side is just a reminder of what the structure of the retina looks like. And the thing to remember here is that the important cell type we're going after is photoreceptors, which are labeled in the slide there. And what you can see in this first, in the merged image, you can see where the photoreceptors are and where the RPE, the retinal pigmented epithelia, or RPE, is. And the important thing to know is where we want to get transgene expression is in the photoreceptors and the RPE. And if you go then to the right, to the panel all the way to the right, you can see that we're getting, when we've essentially filtered out the signal from the other components, you can see that we're getting very good transgene expression, this green signal here, both in the RPE and in the photoreceptors. Can I have the next slide, please? So this is a similar kind of slide or a similar kind of study. And what we're looking at here is a little bit closer magnification of photoreceptors. And this, we're actually looking at expression of the transgene being ABCA4, which is the missing gene from patients with Stargardt's disease. It's a very large gene. It's very difficult to get it to express functionally and express well. Lots of companies have had trouble doing this. And what you're looking for here is red. The red signal here is expression of a functional ABCA4 protein. And that's what we're showing. We're showing that when we administer this to the back of the eye, we can get functional expression of ABCA4, which would be curative for Stargardt's disease. Can I have the next slide, please? So again, why do we think that we can do this? Why do we think we can get there? There's a very important market need. Stargardt's disease has no cure at this point. Macular degeneration, dry macular degeneration, there are two forms of it, has absolutely no cure. And the cure for wet macular degeneration or wet AMD is onerous. You need an injection in your eye about once a month. I don't know about, I mean, I at other conferences, I've asked for a show of hands who would sign up for an injection into their eye once a month. I've never had anybody raise their hand for that one. There's a validated target and a validated cell that you know you need to get expression in. We've done that. We can deliver and express in vitro and in vivo. We're still working. We're working our way through the process. The next milestone for us will be showing that we can do this in a mouse model, and after that, a model in non-human primates. Can I have the next slide, please? This is just showing you a little bit more detail on the development plan. We're showing and very confident we can get into human trials by the end of 2024. Can I have the next slide, please? Here's another one that we find pretty exciting, and this is cystic fibrosis, which we're working on. Another disease with no cure, a genetically defined disease, which is one of the most common genetic abnormalities in the Caucasian population, CFTR. And the next slide, please. So cystic fibrosis is a disease that's caused by a number of different mutations in what's called the CFTR gene. And the way it primarily presents is in difficulty breathing. You're not able, the part of the structure or function of sweeping the gunk out of that builds up in your lungs is damaged. And these patients end up about 20 years ago, before a couple of some of the palliative care products that have come out now, these patients used to die in their teens. Now they generally die in their 20s. They more often than not, they'll die from pulmonary problems. It's akin to slowly drowning to death. Can I have the next slide, please? So what's important in cystic fibrosis is you want to make sure you need to be able to get the gene of interest into cells in the lining or epithelium of the lung. And specifically, what you want to try to get the gene into is into the progenitor cells that are deep inside the lining or epithelium of the lung. And what we show here in this first slide is number one, that we can get if animals inhale REVs or our vesicles that are loaded, we can get them very, very deeply into the lung. Can I have the next slide, please? And that when that starts expressing a particular gene, and in this case, it's just a reporter gene. One back, please. What we show here is that we get excellent expression in the lining, in the respiratory epithelium. And it's actually very deep and all the way through that. And this particular slide is what excited the Cystic Fibrosis Foundation so much and caused them to jump over into our.
Is the gene delivery method always the same?
Pardon me? It's always with our extracellular vesicles. Yes, it's always the same. And so what you see here is very good transgene expression throughout the pulmonary epithelium. Can I have the next slide, please? And this is one that I really like. There are a lot of companies, because cystic fibrosis is so common, there are a lot of companies working on cystic fibrosis. And a company you've probably heard of named Moderna is actually working on that as well. And it's rare to find biotech companies in situations where you can compare yourself head to head with another biotech company. But Moderna recently published data on their cystic fibrosis program. And it's a little different because they've stained the cells a little differently than we have, and the magnification's maybe a little bit higher. But what you're looking for in the comparison on the Moderna side, what you're looking for is darker cells, cells that are stained kind of more black. And this is what they published. So, you know, this has got to be the best thing they've got. And you see some transgene expression there, but it's not very even and it's not very regular throughout the epithelium. Looking at ours over on the left-hand side, you're looking for red. And you can see here that we get excellent transgene expression all the way across the epithelia. Really covers very well. Can I have the next slide, please? And so again, why do we think we can do this in cystic fibrosis and pathway in the lung? There's an important market need. Cystic fibrosis is a very important disease in the United States, and there's still no cure for it. There is very clearly validated target, protein to deliver, and cell type you need to deliver to. We've already shown that we can deliver and express in vitro and in vivo proteins in the lung. Our next milestones here are actually the animal model for pulmonary diseases is actually not a mouse, it's a ferret, believe it or not. So the next milestone for us there is expression in a ferret. We'll move from that again to non-human primates as well. Can I have the next slide, please? Trying to help you get back on schedule.
About 3 minutes.
Oh, okay. Well, then I haven't helped you. Sorry. We believe there will be in the clinic in mid to late 2025. Can I have the final or the next slide, please? It's manufacturing, like I said, is very, very easy and very simple. We can go from raw purchased red blood to loaded extracellular vesicles in about a week. From a single unit of human blood, we believe that we can produce, if we're talking about dosing the back of the eye, we believe that we can produce enough to treat 500 to 1,000 patients from one unit of human blood. So that's the one bag when you donate, which is really incredible. That's probably 3 to 4 orders of magnitude more efficient and less costly than either the viral or the lipid nanoparticle systems there. Can I have the next slide, please? Here's the team. The most important person here probably to point out is Harvey Lodish, who's one of our scientific founders. Anybody who studied molecular or cellular biology probably used his book. He's also one of the founders of Genzyme and Millennium and Rubius and 3 or 4 other companies that are in stealth right now, very active still at MIT. The rest of the team, the senior team especially, are all experienced drug developers. Next slide, please. So we are raising our A round. It's only $25 million. We have already not only got 12.8 committed, but we've actually closed on the first 12.8. 5 of that has come from the Cystic Fibrosis Foundation, other investors as well. And I think that's it.
Was cystic fibrosis diluted?
Pardon me? Yes, it was. So Cystic Fibrosis Foundation used to do grants. They have now switched to doing all equity investments. And I think that's the last slide. So time for a few questions. Who? Nope. Maybe I should talk to him. It's the typical, what I'd call the typical R&D collaboration in biotech. So it's not a JV. Yes, exactly. Yes, sir. The Series A, we believe the Series A will get us certainly up to the IND for Stargardt's and probably close to an IND for macular degeneration right afterward. The macular degeneration program we recently have added a guy at Yale, Brian Hafler, who's fairly well known neuro-ophthalmologist at Yale, and the focus of that work with him is going to be on macular degeneration. And it's pretty clear. I mean, one of the things we've shown is if we can express one protein fairly consistently and fairly well in a cell type, we've already shown that if we can do it with one, we can do it with nearly anything. Sure. Yep. Sure. So a couple of things. One thing to note, and that I didn't know when I was talking about why we use red blood, is red blood has been used therapeutically for over 200 years. The first journal article about it is actually from 1818 in the British Journal of Surgery, I think, at that point. Red blood is, first of all, it is the most, red blood cells are the most abundant cell type in the human body. And red blood cells are probably the best understood cell type in the human body. They lack nucleus, they lack organelles, they lack their own DNA. They're essentially just big bags of hemoglobin. Red blood, there are a number of different red blood products on the market right now, and products that are derived from red blood. And so our needs for those, for addressing markets, for example, like Stargardt's or even macular degeneration, are actually significantly lower than the needs for the folks that are using red blood, like CSL Behring uses red blood and takes donated red blood and makes a number of different products from that. And our needs for that are, for the red blood are probably orders of magnitude less than somebody like CSL Behring. So that's a long-winded way of saying we don't see any problems at this point dealing with this. That is a topic of significant debate right now within the company. The likelihood is that we will probably contract that out. It significantly reduces the amount of capital that we need to bring into the company to establish our own manufacturing suites, our own quality system, et cetera.
Don, you're going to be around all day, right?
Yes.